Hello friends, welcome you all in this session. I am BP Shah, PGT Bio, Kendriya Vidyalaya Keshapuram. Today I am going to discuss the topic biomolecule. It included in chapter 9 of class 11th NCERT biology book. Before going to start the chapter biomolecule, I have asked some question in my previous session, that is respiration 5. That questions are, now I think you all are able to answer these questions. First of all, have a look that questions which I have asked in my last session. What are the net gain of ATP molecule per hexose during aerobic respiration? My second question was, which process make direct use of oxygen? The third question was, the last electron acceptor of electron transport chain during the oxidative phosphorylation is. Next question was, before combining with substrate, oxaloacetate pyruvate changes into which molecule? And my last question was, number of glucose molecule required to produce 38 ATP molecule under aerobic respiration is dash. Now, these are the answer of last session question. Now, I think you all have aware with this question. The answer of these questions are, what are the net gain of ATP molecule per hexose? Hexose, it is a carbohydrate, hexa means 6, which is made by carbon 6 molecule during the aerobic respiration. The answer is 36 ATP. I have discussed about 36 ATP and 38 ATP. 36 ATP is for in that particular cases when the acyl coenzyme enter into the Krebs cycle, that take 2 ATP. So, total ATP formed during the cellular respiration, that is 38 ATP. We can subtract 2 ATP in that particular cases, which is consumed when the transfer of molecule from the cytoplasm to mitochondria, that take 2 ATP. So, total gain in aerobic respiration is 36 ATP. Now, my last second question was, which process make direct use of oxygen? I have already discussed two type of respiration is there. In one, there is no use of oxygen, that type of respiration is called anaerobic respiration. But the question is that which process makes direct use of oxygen? That is, answer is electron transport chain. We have seen in oxidative phosphorylation, the in terminal oxidation, the last stage of the cytochrome A3 that use oxygen, electron and proton for molecular oxygen. So, the oxygen used by electron transport chain, answer is electron transport chain. Now, the third question was, the last electron acceptor of electron transport chain during the oxidative phosphorylation is. We have already discussed the oxidative phosphorylation or the electron transport system, it is a chain of electron carrier as well as cytochrome. The answer was cytochrome A3. This cytochrome A3, it is found at the terminal end which accept oxygen. So, the answer is cytochrome A3. Now, the fourth question is, before combining with substrate, oxaloacetic acid या oxaloacetate pyruvate change into, mean in Krebs cycle which molecule combined with the OAA या oxaloacetic acid, oxaloacetic acetate, because the pyruvate can't enter into the mitochondria from the cytoplasm. First of all, pyruvate is going to be changed into which molecule? The answer is acetyl coenzyme. Pyruvate changes into acetyl coenzyme. Now, acetyl coenzyme are able to enter into the mitochondria and combined with oxaloacetic acid which form citric acid. Now, my last question was number of glucose molecule required to produce 38 ATP under anaerobic condition. This is very important point. This is the case is anaerobic respiration. We know in anaerobic respiration that total number of ATP liberate, that is 2 ATP. So, one glucose molecule liberate 2 ATP. You can solve by this way. In anaerobic case, one glucose molecule it gives 2 ATP. So, here the answer is how much glucose molecule is used to form 38 ATP. So, one glucose molecule 2 ATP, 19 glucose molecule gives 19 into 2, that is 38 ATP. So, in case of anaerobic respiration, when we use 19 molecule of ATP, that gives 38 ATP. So, the answer was 19 glucose molecule. If it is the case of aerobic respiration, then there is no doubt the number of glucose molecule used to produce the 38 ATP in aerobic case, that is 1 glucose molecule. Okay. So, after this I hope you all will be able to answer all type of questions related to the cellular respiration. Okay. Now, let us start the new chapter biomolecules. It is very important for your examination point of view as well as its application in class 12th, because in class 12th there is a chapter called molecular biology. In that cases all the biomolecules are used in that particular cases, either in the DNA formation, RNA formation or various types of amino acid is formed in case of the genetic code. So, this is very important and it is also related with the co-linked with the class 12th. So, let us start the chapter biomolecule. What do you mean by biomolecule? As the name indicate, the molecules and bio. Molecules, the different types of atom combined in a specific ratio. That specific ratio gives molecule. Just for example, one atom of carbon and 2 atom of oxygen, when combined this form carbon dioxide. The carbon dioxide is molecule and carbon and oxygen is atom. Bio, this molecule when present inside the living cell, living cells, that molecule is called biomolecule. So, the molecule which are present in living cell, living organism, that are called biomolecules. Now, we can easily define, there are in periodic table you have seen various types of atom are present. There are many 118 known elements is there. Out of 118, 25 has been identified. These are present in the cell or extracellular fluid, that is called ECF, that are called biomolecules, biological element or bioelement. So, we can define in some way, that is various type of molecules found inside the cell for the purpose of different physiological, morphological, cytological purposes, that are called biomolecules. These molecules help in the various process, these help in the various types of cycle like Krebs cycle, electron transport chain, like Kelvin cycle. These biomolecules is used and complete the cycle. Okay. So, this is the definition of simple definition of biomolecules, the molecules which are present in the living cell. Now, these biomolecules has been classified into two main group on the basis of availability in the cell. These biomolecules are known as macromolecule and micromolecule, MICRO molecules. As the name indicate, macro means small, macro means MACRO means large, macro means small. So, simply we can say those molecules which are present in large quantity in the cell or in the living organism, that are called macromolecule. Those which are present in the small quantity, that are called micromolecules. Now, what are the amount in that particular cases? So, biomolecules are classified into biomacromolecules, biomicromolecules. Biomolecules which have molecular weight less than 1000 Dalton, or we can say excess of 10 mmol per kg of the dry matter, and are usually referred as biomacromolecule. So, those molecules which are present in excess of means greater than 10 millimole per kg dry weight, dry matter. So, its quantity in the living organism must be greater than 10 mmol per kg, but when the amount is less than 10 millimole per kg dry weight or dry matter, that is called macromolecule. So, we can easily define that biomolecule which have molecular weight ranging from 18 to around 800 Dalton, less than 10 mmol per kg of dry matter, that is called biomicromolecule. So, we can easily differentiate in between the biomacromolecule and biomicromolecules. So, biomicromolecules greater than 10 m mole per kg, when it is less than 10 m mole per kg of dry matter, that is called biomacromolecules. Now, the entire biomolecules according to the your syllabus has been divided into 4 groups, that is carbohydrate, lipids, proteins and nucleic acid. Nucleic acid, which is the DNA and RNA, deoxyribonucleic acid and RNA ribonucleic acid. These are the 4 major categories of the biomolecules. First of all, carbohydrate. Carbohydrate, as the term indicate, it is the hydrates of carbon. We can simply define carbohydrate, it is the hydrates of carbon. Hydrate means water. When water combine with carbon, then it make carbohydrate. So, simply we can say the carbohydrate are the biomolecules which are made by carbon, hydrogen and oxygen, in which the hydrogen and oxygen ratio is always 2:1. There are various types of carbohydrate. Here in case say, if the general formula of carbohydrate we can write CN H2N ON. Here you can see 2N is hydrogen, N is oxygen. So, the ratio is 2:1. In carbohydrate, the hydrogen and oxygen ratio always will be 2:1. Okay. Now, there are different types of carbohydrates are there. The carbohydrate which serves as a building material. Now, types of carbohydrate. Now, there are different types of carbohydrates are there. We can classify it monosaccharide, oligosaccharide, and polysaccharide. So, carbohydrate can be classified on the basis of number of carbon atom or on the basis of number of monosaccharide. We can differentiate the carbohydrate into these 3 types. First of all, monosaccharide. Monosaccharide are those type of carbohydrate which after splitting it gives 3 to 7 carbon atom. So, we can simply say that the general formula of carbohydrate is CN H2N ON. When N varies 3, 4, 5, 6, 7. So, that type of carbohydrate is called monosaccharide. Now, oligosaccharide. Oligosaccharide, when oligosaccharide is going to be split, it gives more than 2 monosaccharide. On that basis, the monosaccharide has been classified into different group. One is called disaccharide. As the name indicate, di means 2. When disaccharide is hydrolysis takes place in disaccharide, it is split to form 2 monosaccharide. When it gives 3 monosaccharide, that is called trisaccharide. When the trisaccharide is going to be split, it gives at least 3 monosaccharide. So, oligosaccharide has been classified into disaccharide, trisaccharide, tetrasaccharide, and so on. So, we can define the oligosaccharide when the oligosaccharide is split, it gives 3 to 9 monosaccharide unit. In disaccharide 2, it is simply called disaccharide, and trisaccharide, tetrasaccharide, pentasaccharide, it combinedly called oligosaccharide. So, carbohydrate first is monosaccharide, second one disaccharide, third one oligosaccharide. Oligosaccharide gives 3 to 9 monosaccharide, disaccharide gives 2 monosaccharide, and lastly polysaccharide. After hydrolysis, when the carbohydrate gives more than 10 monosaccharide, is called polysaccharide. So, now we are able to say the carbohydrate has been classified into 4 main category, that is monosaccharide, disaccharide, oligosaccharide and polysaccharide. On the basis of number of carbon atom, on the basis of number of monosaccharide, that classification is best. Now, first of all, one by one, this is very important for your examination point of view to see the structure of different types of monosaccharide. You can easily make this type of structure. So, first of all, monosaccharide. Monosaccharide, it always, as we have already discussed, after splitting, after hydrolysis, it gives 3 to 7 carbon atom. And the formula is CN H2N ON. Now, when the monosaccharide gave 3 atom, yeah, made up of by 3 atom of carbon, that is called triose. Triose, that is C3, the formula is, here the N is 3, hydrogen will be, okay, very good, 6 and oxygen will be 3. So, the general formula of triose is C3 H6O3. Now, its structure depend upon the carbonyl group where the carbonyl are present. It has been divided into two main part triose, that is aldehyde and ketone. Aldehyde we have already know its functional group is CHO, the ketone functional group is C double bond O. Now, the example of triose in aldehyde group, that is glyceraldehyde. At the time of writing the formula of structure formula of glyceraldehyde, we know that glyceraldehyde have 3 carbon. So, it will be easily write C, C, C. These are the 3 carbon atom and 1 carbon atom has CHO group. So, here the CHO, that is the aldehyde group. Now, here H and OH. At third position of the carbon, it is CH2OH. So, we can easily write the formula or structure of the glyceraldehyde. CHO at one position, in second position H and OH, and the third position CH2OH. Now, you can count that carbon is 3 C3, hydrogen 1, 2, 3, 4, 5 and 6, hydrogen is 6, oxygen 1, 2, 3, oxygen is 3. So, we can easily write the formula of triose or the glyceraldehyde, which is a aldehyde group and the structure of this type. Now, the second form of triose is ketone. Here we can easily write the general formula will be the same, because it is triose. Triose general formula is C3H6O3. Now, the structure will be slightly different, because here is the COO group, ketone group are there. So, we can easily write C3 carbon. Okay. At second position there is carbonyl group, and in the uppermost first one there is CH2OH and the third one position CH2OH. So, this is the formula of ketone triose. Its name is dihydroxy acetone. You know in class 10th chemistry, this is the acetone, have functional group COO group. Now, you can verify the formula, it is true or not. Here carbon is 1, 2, 3, that is C3, hydrogen 2, 3, 4, 5 and 6, hydrogen is 6. Now, oxygen 1, 2 and 3, oxygen is 3. So, this is the formula of triose. Two examples, that is the glyceraldehyde and dihydroxyacetone phosphate, dihydroxyacetone. These two compound are easily used or in photosynthesis chapter as well as in respiration chapter. So, you think and write the formula of triose. Next carbohydrate, that is tetrose. As the term indicate, tetra, tetra means 4, here the number of carbon is 4. Now, the formula will be C4H, okay, very good, 8 and O will be 4. So, the formula of tetrose is C4H8O4. Now, it is also found in 2 form, same as triose. Now, you can easily write two form are very good, that is aldehyde and ketone. Aldehydic form that contain CHO group, ketone form just we have already discussed in this group. Okay. Now, you can easily write the structure formula, structure of the tetrose monosaccharide. If the carbon 4, you can easily write 1, 2, 3, 4. These are the 4 carbon. We have already discussed at 1 carbon in first position there will be the CHO group. Aldehyde group are present at the first position. Okay. Next one you can easily write HOH, HOH, and last one same as triose, that is CH2OH. So, this is the structure of tetrose, that is called erythrose. This also used in photosynthesis, that in Calvin cycle. The another aldehyde which are frequently used in biochemistry of tetrose, that is triose. Triose is a tetrose, the formula will be same, that is carbon 4, you can easily write 4 1, 1 2, 3 and 4. Now, we have already know that triose is a type of aldehyde. So, the CHO group will be found at position 1. So, this is the CHO. Now, you can easily write the in second position the HOH will be going to be changed. In OH group are found in the left side and H is found at the right side, and same as erythrose third position have HOH and the fourth one have CH2OH. So, we can easily write 2 tetrose, erythrose and triose. Here the number of carbon you will find 4, 1 2 3 4, 1 2 3 4. Hydrogen 1 2 3, sorry, oxygen 1 2 3 4, hydrogen 1 2 3 4 5 6 7 and 8. So, our structure is correct, you can easily found in the triose, same will be there. Now, come to the ketone. The ketone form you have already know how write the tetrose of ketone, that is example here is erythrose, erythrulose. There is a similar term like erythrose and erythrulose. Erythrol is a lose is a ketone and erythrose is a aldehyde. Now, you can write the structure of erythrulose, that is carbon 4 1 2 3 4. Okay. Now, in which group carbonyl group are present, CO group are present, that is 2, very good. At 2 position carbonyl group is present, CO group. So, at 1 position CH2OH and same as erythrose or triose, third position H and OH are present, and last position same, that is the CH2OH are present. So, we can easily write the structure of triose and structure of tetrose, their formula in triose C3H6O3. It is also found in the 2 form, aldehydic form and ketonic form. Example is glyceraldehyde dihydroxyacetone. In tetrose the formula is C4H8O4 and the examples are erythrose, triose and erythrulose. Erythrulose is a ketonic form and erythrose is a aldehydic form. So, these are the two forms of monosaccharide, that is triose and tetrose. Now, we can easily write the next monosaccharide, that is pentose. As the term indicate, the penta, it is made by penta. Penta means 5. Here the carbon atom 5 and the formula will be same, Cn H2n ON. H will be very good 10 and O will be 5. These pentose sugar are very important in the molecular genetics, because in molecular genetics the DNA and RNA consist of pentose sugar. This DNA contain deoxyribose sugar and RNA contain ribose sugar, and the example will be the same. In case of pentose, it is also found in the two form same as triose and tetrose, that is in aldehydic form and in ketone form. Ketone form aldehyde form CHO, ketone form C double bond O, the two alkyl groups are present. Now, the pentose, the aldehydic form of pentose, example ribose, it is very important, ribose. Now, you can easily write without any hint. Ribose is a 5 carbon monosaccharide. So, you can write 5 carbon 1, 2, 3, 4 and 5. These are the 5 carbon, no doubt. Now, at 1 carbon what will be present, that is CHO group, and in next carbon same as triose and same as tetrose, it will be write as HOH, in third position HOH, in fourth position HOH, and in last position what will be here, what will be there, that is CH2OH. So, you can easily write the structure of ribose. It has 5 carbon 1, 2, 3, 4, 5, hydrogen will be the 10, that is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10 hydrogen, and oxygen are 5, 1, 2, 3, 4, 5. You can easily write the structure of ribose. This ribose, and now come to the ketone. The ketone, the examples are ribulose, the ribulose, here the carbonyl group are present in the form of ketone. Now, the structure of the ribulose will be, it contains 5 carbon. So, you can easily write 1, 2, 3, 4, 5. In which position COO group is there, that is at second position. So, we can easily write at first position, that is CH2OH. Now, it is same as ribose HOH, HOH and CH2OH. So, we can easily write the structure of ribulose. Ribulose is a ketone, pentose ketone. Now, the next important pentose, that is deoxyribose. Deoxyribose is a type of aldehyde, but the name indicate deoxy means deletion of one oxygen from the ribose sugar. Ribose sugar is a pentose sugar. At one carbon atom, one oxygen is going to be deleted. This carbon atom from where the oxygen is going to be deleted, that is carbon 2. If we will know these things, there is a deletion of one oxygen at carbon 2 from the ribose, we can easily write without any problem the simple structure of deoxyribose. That now start. Deoxyribose, it contain 5 carbon 1, 2, 3, 4 and 5. Now, it is a type of aldehyde, no doubt. So, at position 1 CHO group, at second position we already know the deoxy, one oxygen is going to be deleted from the ribose sugar. In ribose sugar, here the H, OH are there, here O is deleted, only H is here. Now, same as ribose, H, OH, H, OH. At 6th position, what will be there? That is CH2 OH. So, we can easily write the formula of ribose, deoxyribose and ribulose. The ribose are the pentose sugar which are used in formation of RNA, and deoxyribose are the pentose sugar which are used in formation of DNA. On that basis, its name are there RNA, ribonucleic acid. Ribo means in RNA ribose sugar are present, in DNA there is deoxyribose sugar are present. So, the name is deoxyribonucleic acid. But one thing is here is very essential, these pentose sugar are not present only in the simple chain like straight chain, it is found in the form of ring structure also. The ring structure is also very important for the structural point of view. In the formation of DNA, when you write the structure of DNA, you may require the ring form of pentose sugar, that is ribose and deoxyribose. Now, the ring form of ribose sugar you can easily make it. Here is the O, oxygen is here, here is the carbon, here is also carbon, carbon. This is the skeleton of ring form of the pentose, that is ribose, ring form of ribose. In ring form or straight form, the structure formula will be the same, that is C5 H10 O5, but how you can write? In these cases, here OH group is here and H group is here. Here at the second position, this one is the one position, at second position, this is the third position, this is the fourth position and fifth carbon present here, this is the fifth position. At second position in the straight form, this is the H, OH. At third position, it is also H, OH. Here is H, CH2OH. So, we can easily write, when we know the straight chain structure, put here at one position OH, H, H, OH, at third position also H, OH and the fourth position H and CH2OH above the 4. Now, when we understand the ring formula of ribose, we can easily write the structure of deoxyribose sugar. What will be the structure of deoxyribose sugar? Deoxyribose. Formula will be, in deoxyribose sugar one oxygen is deleted. So, the formula will be C5 H10, instead of O5 here is O4. So, now write the structure. Skeleton will be the same like the ribose, C, carbon here, same as ribose. Now, when we know that at second position of ribose one oxygen is deleted, then the ribose for the deoxyribose. That means, it is the one position same OH, H here H, at this position oxygen is going to be deleted, at second position oxygen are not present. So, we can write H and same at third and in fourth same as ribose, here is H, here is CH2OH. So, now we can easily write the difference between the ribose sugar and deoxyribose sugar, that is ring structure form and the open chain for ribose and deoxyribose. When we know the open chain form, we can easily write with the help of skeleton of this O position here, oxygen is here, carbon, 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 then we can easily write. When you write the structure of ribose and we know the difference between the ribose and deoxyribose, at which position oxygen is going to be deleted, that we can easily write the structure of ribose as well as deoxyribose, which is the main sugar present in the DNA, which acts as a genetic material of living organism. Okay. So, this is all about the pentose sugar. Okay. Now, the next will be the hexose. As the term indicate, hexa, it is made by hexa means 6. It contains 6 carbon. When we know the formula of carbohydrate, that is CN H2N ON, we can write easily the formula of hexose. What will be the formula of hexose? The carbon will be 6, okay. Hydrogen will be 2N, means 2 into 6, that is 12. Oxygen will be N, that is 6. So, the general formula of hexose is C6H12O6. Okay. Now, this same as triose to pentose, these hexose are also found in the two form, that is aldehydic form and ketonic form. Aldehydic form, it contain CHO group, no doubt, same as triose, tetrose and pentose, and the ketonic form, it contain ketonic group, 2 alkyl groups are there and CO group. Now, the aldehydic form of hexose is glucose. Glucose is the main fuel of the living organism. It is used in the cellular respiration as a chief fuel, which going to be oxidized and form carbon dioxide, water and ATP. These ATP are used various metabolic, various physiological, morphological changes. So, glucose structure, that is the it contain carbon 6. So, we can easily write the structure of glucose. In glucose how much carbon? Okay, that is 6. So, you can write 6 carbon in linear way 3, 4, 5 and 6. Now, what will be the position of CHO? At 1 position, and what will be the HOH position? In second position, hydrogen left side, OH this side. In third position, the hydrogen here and OH is here, and same H, OH, H, OH. Now, at the sixth position, CH2 OH. So, without any problem you may write the structure of any type of monosaccharide, either it is triose or tetrose or pentose or hexose. So, now you all are able to write the any type of formula if you know this structure, either aldehyde or ketone, and the position of ketone and position of oxygen and position of H, hydrogen and OH group, you can easily write the whole structure that can be utilized in your another chapter like genetics, like physiology, photosynthesis and respiration. So, this is the one example of the aldehydic form of hexose. There is another example of hexose we can largely use in biology, that is galactose. That is also known as milk sugar. The galactose, when we know the galactose is a type of hexose, the formula will be C6H12O6, then you can easily write 6 carbon 3, 4, 5 and 6. When we know the galactose is a type of aldehyde, so we can easily write the CHO group are present at position 1. So, you can easily write CHO, then the position of H and OH, position of H and OH from the glucose, when you know where the position can be changed, that you may write that in second position same as glucose H OH, at third position same as glucose H OH, but the difference at carbon 3, here in glucose H is left side and OH is right side, but here is same as second H OH, here H OH, here CH2 OH. So, this is the aldehydic form of hexose, that is two example here, one is glucose, another is galactose. So, now we can summarize whole things, that is carbohydrate are found in monosaccharide, disaccharide, oligosaccharide and polysaccharide. Now, the structure of triose, tetrose, pentose and hexose. Now, you can easily think over these questions. What are the other name given to the carbohydrate? As I have already discussed many times in carbohydrate, that is the other name of carbohydrate you can find. My next question is, what is the common sugar found in animals? My third question is, what are macromolecules? Give examples. Thank you and prepare for the next section. You can easily write everything with the help of your previous knowledge. Thank you.